Okay, sounds good. Yeah. See, okay, I have a spare, you pick up. They kind of. Oh. It's just like, you know, the same. We tried this over there. Oh, yeah. I just take a while. My parents have been, they use my. Okay. I'm rather like some low. You can do it like you can make the light on.
Oh, okay. Hold on. I need to watch my dad.
Change slide, Eric can help you, you know, you can see here.
Okay.
Release about board Thursday, right? Thursday. Today is your second day here. Yeah. This our summer research program student. Yeah, last time she didn't have the chance to do the presentation, so we give her 10 to 15 minutes today to do your presentation. Okay. Aren't you using brain? No, we haven't that they have. Yeah. Okay, why don't we just talk?
Okay. Okay. Hi, my name is Elizabeth. I'm 17 years old and I'm an upcoming senior in high school. I used to live in San Gabriel, but a few years back, my family and I moved to Texas, in Dallas, that area. So that's where I go to school, but every single summer I come here and I live with my grandparents in Alhambra. And so this is my family. This is my mom, my grandparents who I'm living with. And then this is my dad and my brother. This is from when we went on a hike in Sequoia. We really like hiking. And so just to talk about my family a bit, my family is really, really important to me. They've always told me to work hard and to do things in life that make me happy and to pursue things I'm passionate about. So I'm really thankful for everything they let me do. And then just like a fun fact is I'm a self-proclaimed gingerbread housemaking champion. Every single Christmas, my friends and I get in groups and we make the houses and I've yet to lose. So that is a fun fact for you. Let's go on. Okay, so just some hobbies of mine. I think my longest hobby is art. I've been doing art since I was young, and I love it because it's a really good creative outlet for me to put different ideas together, do sketches, and just put everything together into one thing that I want to portray and put out there. And I also see art as being more technical than most people realize because it takes a lot of attention to detail and spatial awareness of how things go to make art. So these are just some of the art pieces I've made and won awards for. So this is a cat I did, and then this is a painting of a waterfall from a hike that I went on with some family friends a few years back. And then this is a scene from a movie you guys may know called Parasite, which won the Picture of the Year at the Oscars a few years back. And so it's one of my favorite movies. After I watched it, I went down this whole YouTube spiral about the behind the scenes and the hidden meanings. So I decided to do a scene from that movie. Yes.
Very nice.
Yeah. So, and then another hobby of mine is reading short stories. I had this really amazing history teacher this year who while we learned US history, would give me recommendations for short stories that went along with whatever time period we were learning about in school. So for example, he had me reading like Edgar Allan Poe, James O'Brien, modernism, postmodernism. So it became a hobby of mine to read these stories. And I just wanted to share a few, or just one with you. This is my favorite one, but I wanted to talk about this science fiction one called The Toxic Donut. One, because it's kind of a bizarre story, and two, because I've heard that you guys like donuts around here. I even took one this morning. And so The Toxic Donut is set kind of 200 years in the future. And it's kind of during this end of the year award show just to celebrate the new year. And there's this guest of honor who is honored the whole night. And at the very end, this guest of honor has to eat this toxic donut. And this donut is basically humanity's solution to environmental issues. So every single year, they take all the toxic waste and pollution in the world and compile it all into this one donut that this guest of honor, or in other words, sacrifice, has to eat and take one for the team. So it's a really crazy story, but it was written kind of during the 90s when a lot of environmental groups were rising during that time period. Another hobby of mine is photography. I love photography, whether it's while traveling, hiking, or for a bunch of school clubs I'm a part of. I do a lot of the social media for a lot of my school clubs. For example, I am the historian for this program I'm in at my school called Carroll Medical Academy. And I'm just grouped with other kids who are interested in the medical sciences to take classes, not just biology and chemistry, but like medical terminology or anatomy and physiology. And we take these classes together, and my job as historian is to take pictures at all the events and put these events together. And I also, completely on a whim, like a year back, I decided to put some of those pictures on Pinterest. And then one day I woke up and my phone was blowing up, and some of them kind of blew up on Pinterest. So I have around like 45,000 saves, and then almost a million combined views from all those photos. So that's kind of something bizarre that kind of happened to me that I'm kind of proud of. And my last hobby is going to museums. I love going to museums, whether it's art museums, history museums, or science museums. And I love just driving either by myself or with my friends to go. Involvement, just some things I'm involved with after school and in school. I'm part of the dragon dance team at my school. I originally just joined and I was not expecting to love it so much, but it was really, really fun. Still part of it, and there's just some pictures of us. Think Green is another club I'm really involved with at school, and it's an environmental club. And some things we've done this year are we started a recycling project at our school where we set up bins and posters and have a whole system of running it. This is just a poster from one of the meetings. And then we also took old Capri Sun bags and we sewed them into containers for our teachers to use to hold school supplies. So that's kind of some fun things we do with that. And I love that club. And then another big time commitment I have outside of school is I work tutoring jobs, probably 8 to 9 hours a week. And it's really, really rewarding for me. I work with kids younger than me and with families, and I love seeing them get better, and just, I love working with the kids. So yeah. Just some more interests. My favorite movies are The Menu and Good Will Hunting. If you guys have ever seen Glass Onion or Knives Out, those kind of movies, The Menu's, I would say, a bit of a similar vibe. And I'm also a huge foodie, so liking The Menu makes sense. If you guys have ever watched it, I really recommend it. I like music of all genres. I'll listen to pop, indie, a little rap, a little K-pop, a little country, everything. But if you ever tap me and I have earbuds on, I'm most likely listening to Taylor Swift. And my favorite album of hers is Red. And it's the first album I ever owned physically. So I love that album. And then another thing is I like watching sports with my family. These are just a few teams that my family and friends and I watch. And why this program and my goals. So I took AP biology as a freshman, which was also during COVID. So I had to do online school that year and I wasn't able to get exposure to many labs. A lot of my labs were done online through like programs. And so I was really hoping to find a program where I could get really hands-on lab experience and develop those lab skills. And like learning problem solving, teamwork, collaborating, and all that was something I really wanted to get more involved with. And then also I loved learning about biology, genetics, and chemistry in school, but I wanted to take that next step and learn how to apply these subjects I like into like a project or seeing how they work in different processes that we use in the lab. And then also this program really special because it offers the idea to work with a mentor, which is really special because it's great to have someone to have guidance and support in doing something I've never done before. This is my first experience with research, and it's really special to have that relationship and guidance. And then lastly, kind of more about my goals and what I want to do. I know I want to get involved with the medical sciences in my future. I actually had the opportunity last summer. I shadowed some physicians, and that only solidified that I really wanted to be in these fields somehow. And I'm here because I really want to get exposure to the research side of things, see what goes on behind the scenes of the development of these life-changing medicines, and get exposure to biotechnology. And I'm hoping that what I learned here and this experience I can use and contribute to my future and what I want to do. Like that. Yeah.
Thank you. Any questions for you?
Questions?
You say you like to read short stories, so you know your teammate.
Oh, really?
You're very good at copying.
Now we can have a competition by the end of the now. Or we can work together to come up with a short story.
Yeah, we
At the
can-
end.
Good. Very good. Not only you can contribute to the story, but you can do the artwork also. For the stories you can publish together.
Yeah.
What is the program you are now in school?
It's like kind of a track where like, oh, you have to take bio freshman year, and then you have to take med term this year of high school, and then we just take them like with the same group of like 60 people into 60 people throughout each year of high school, and you kind of work with them and you do like dissections and that kind of thing.
Okay, great, thank you. Okay, now next we will have, let me turn off this, and next we'll have Josh to give us the journal club presentation. Thank you.
I can't really find it. I have it sort of.
Yeah, your good presentation, I put it by him, so maybe you have to click and.
Oh, sorry.
Hmm?
I saw that.
You got it?
I found that.
Yeah, go to the link. Watch meeting. All right, John. Have that meeting. Need to then you mute your computer and you can start. Okay. I do have to go there, share your screen, stop sharing, and you can share your screen. Okay, we got. You share your screen. You share screen.
Yeah, nice presentation, Elizabeth. I also like Good Will Hunting. It's a good movie. So I followed George's suggestions, and basically I'm gonna also give you some like of my background and what are my interests as well. So after that, I'm gonna basically focus on something I've studied before in the past, which is on it involves a little bit of biochemistry, chemistry, and it's basically it involves very special enzymes, and I'm also gonna relate it how it ties into cancer. So more or less going forward, I was born in SoCal. My journey through academia really took me everywhere in the US, a lot of places in the US, I should say. And yeah, in many ways, my taste in things are also pretty widespread. So I was born in Long Beach, California, at Long Beach Memorial Hospital. My hobbies include dog walking, trying new spicy foods, and listening to music. And so in the lower left corner is my dog, who I named after Chewbacca. I call him Chewie. So he's a very happy dog. Most of the time we spend a lot of our time hiking through Santa Monica Mountains or along the mountains. And here goes a picture of my friends that I took right before the pandemic hit in New York City. So I went through school for quite a while. Mainly my studies have been on biomedical sciences and biochemistry. Started off studying biochemistry. Eventually I ended my academic career at NYU with a master's degree, and eventually started working at Amgen for a year, helping them develop antibodies in Thousand Oaks. And in 2022, and it's actually getting very close to my anniversary date, I started working at Therogen under the R&D team with Venki and John. And now we recently had Sergey join us. And so now focusing on the science behind my presentation, basically I'm going to present on two different subjects. One is on reactive oxygen species, and I'll go into a moment what that means and its relation to cancer. And then more or less, like I had someone ask me a question about an enzyme I studied and whether or not it's like found in this like model organism called Stentor cerealis. And I'll go into like what that species actually is, and then I'll go into the conclusions. So more or less, what really got me interested into chemistry and science is that I've always been amazed at proteins and how proteins can be engineered in so many different ways. So amino acids basically form the backbone of proteins. Like one amino acid comes together with two amino acids through a process called condensation, and basically that releases water. But amino acids react enzymatically within a cell to form proteins, and they can be arranged in so many different ways. And you can basically model this like through a permutation if you wanted to show that it can form like things like hemoglobin or a receptor. So there's always been this adage in biology that structure determines function. And to understand function of the so many myriad of proteins that perform vital jobs in a healthy body or malfunction in a disease way is something scientists have tried to fathom for a very long time. And so that I thought really got me interested in structural biology when I was really young. And eventually this path led me to immunology and cancer immunotherapy. And so this is what initially got me started off in and interested in science. And so the next thought that came to my mind and what I really have been interested as of recently is how can we re-engineer like proteins to help the body fight disease such as cancer. So proteins form vital jobs. They drive to fuel the body with ATP to produce energy for the cells, but at the same time, they're also producing reactive oxygen species. And some cells do it more so than others. Especially some immune cells like macrophages, they produce NADPH oxidases that are known to create superoxide. And so like some cells are more equipped to better fight reactive oxygen species better than others, whereas some aren't. And it goes into questioning is like how do some cells do this and how do some cells not do this, and what makes cancer cells so special to combat this. So every day we get about like 1/10 of a million of oxidative hits per day, and our body uses it's been naturally evolved to express or produce certain proteins that can quench or basically nullify the radicals such as superoxide dismutase and catalase. So we have natural enzymes that can help us do this. There's basically an equilibrium within every cell that helps to quench these radicals. And so if the reoxygen oxygen species exceeds what the cell can handle, it basically leads to what they call oxidative stress, and eventually leads to cell death. And basically what people have been studying since about like 2012 is a mechanism of cell death called ferroptosis, where cells are not able to quench or nullify all the reactive oxygen species within a cell. And so that leads to a certain type of cell death. There's different types of cell death, but this is the one I'm going to focus on today because it seems a little bit more relevant today, especially in the field of cancer from what I've been reading. So more or less, some radicals are naturally produced as signaling molecules. So on the left is a balanced equation of how like nitric oxide, which is also a radical, is produced. But this actually occurs naturally within our body by a set of enzymes called nitric oxide synthesis. So basically, nitric oxide synthase is thought to have evolved from a gene fusion event with a long time ago within mammals, where you have a what's like an NADP oxidase fused to a cytochrome Q450. And basically, they think these genes came together within mammals a long time ago to help produce this enzyme that I'm showing you on the far right. So I'm not going to go into the full mechanism. It's a bit complicated, but essentially it involves a redox mechanism where if the enzyme takes electrons from NADPH and will oxidize the amino acid or L-arginine to nitric oxide and produce L-citrulline as a co-substrate in the process. So this is special to mammals, but it's also interesting that like as I mentioned before, they believe that this came through a gene fusion event. We also observe the same thing within prokaryotes, small algae, as well as like extremophiles. So it's a very interesting enzyme that's special to us. And again, it's a free radical, and that's why I incorporated within this presentation. So essentially, our body naturally produces free radicals as messengers or neurotransmitters in this case. So radicals like an are able to freely diffuse across the membrane. These molecules undergo both paracrine and autocrine signaling. So in the body, basically, you have signaling that occurs when cells talk to themselves, and then they also talk to adjacent cells or the nearby environment. So what's special about these like radical molecules is that they're able to talk to both. They're able to talk to themselves as well as talk to neighboring cells and induce some sort of change within neighboring cells. And so basically, NO, which is a free radical, is able to cause neurotransmission when it is enzymatically produced and binds to the adjacent neuron, and basically it binds to something called soluble guanoic cyclase, and that creates a secondary messenger called cyclic GMP. And so, but it's also found within immune cells. So basically, there are three types of these enzymes found within mammals. One is iNOS and nNOS, which is neuronal, and then endothelial cells, which is also responsible for vasodilation. And so NO is also special because it acts to kill cells at times if the cells, again, cannot buffer this oxidative stress. It's been shown that at lower concentrations, it leads to tumorigenesis, but like if you overwhelm the cell with nitric oxide, it leads to cell death. So it could be quite interesting. So every cell in our body produces reactive oxygen species, but some need it, and some cells rely on a delicate equilibrium to quench it before it becomes deadly. So feral apoptosis, as I mentioned to you before, is a type of cell death that's marked by membrane blistering. The mitochondria are basically shown to look dead before the actual cell itself looks dead. So the hallmark of feral apoptosis is that the mitochondria dies, which is like the powerhouse of the cell. And so an inability to quench ROS leads to oxidative stress, oxidation of iron cofactors, which are basically needed in the mitochondria to produce ATP, and it leads to what is known as lipid peroxidation. So basically, they call this a positive feedback loop. So the reactive oxygen species has one free radical. It'll react with another molecule that causes it to also create another free radical, and it propagates essentially. And then this is the mechanism by which it leads to cell death. Our body, again, has natural antioxidants and enzymes that are prepared to quench these free radicals. And so, interestingly, some cancer cells, they have multiple mechanisms by which they achieve immortality. One of the mechanisms is that they take advantage of the lipid environment, or they basically find ways to inhibit feral apoptosis or this imbalance in redox potential. So more on that point, here goes a picture. So cancer cells prevent feral apoptosis by taking advantage of multiple mechanisms, including increasing lipid uptake. So essentially, when cells achieve metastasis, they will migrate to lipid-rich environments such as lymph nodes to increase the amount of lipids in the cell, and basically buffer the reactive oxygen species within the cell. So they take advantage of multiple mechanisms, and they also like to stay out of the blood where it's also iron rich, and which would also increase the amount of reactive oxygen species within that cell. So they don't always like to stay in the blood either, because it's iron rich, which will also increase the amount of free radicals within that cell. And so going back to the point I made before, do reactive oxygen species, or does this occur in other organisms as well? And why don't they develop cancer? And so these sort of enzymes do exist, again, in prokaryotes, eukaryotes, alga. The smallest of these enzymes is found within a green alga called Otari. So basically on the left, I show the enzyme NOS, and then on the right is a picture of this very small photosynthetic organism called Otari. It's about little less than 1 micron. So that's 10 to the negative 6 meters in length. So we're talking about the size of a molecule the other day. That's like 10 to the negative 10, about the size of a molecule. So this is like very small, but it still produces this molecule. And so right below that image is the different kingdoms of life that where NOS is found. And so right in the middle of that, so like basically N terminus basically designates where the protein begins, C terminus designates where the protein ends. So more or less, I'm just trying to show that like this protein is found through all kingdoms of life, which basically shows its significance. But it's only in mammals that we have a gene fusion event and reactive oxygen species becomes a problem. So it's sort of interesting that you can find this protein through all kingdoms of life, which basically usually when that happens, it displays its significance throughout life. So I recently had someone reach out to me, or like I was talking to someone, they asked me like, does NOS also occur within the species called Stentor ceruleus? So Stentor ceruleus is a model organism for regeneration. It basically, if you chop it into two, it can regenerate. It's a very small organism. So I thought that was also interesting that like you can also find this enzyme within a species like that. Like if another area of research too is regeneration. How can like some things regenerate by reprogramming itself? So usually when people find proteins within a database, they deposit it on and what becomes available onto a server. So I found ways to find NOS within this species called Stentor. So it's basically shown on the left. It's called the trumpeter of the microscopic symphony orchestra. It's a very large single-celled organism that very much resembles paramecium. So what's interesting, it's ciliate, so it can move by itself. Again, like just over a weekend, I was like, can I find NOS within this species? And I did. And basically, and this is something I probably can show the students in the future, I can basically use AlphaFold to generate entire complexes or proteins of what's found in this organism and what are considered hypothetical proteins. So hypothetical proteins basically don't have any gene ontology, or like they're not annotated in any way to designate what they actually are. So people think that they don't actually have any function, but they actually might have some function. Usually people see an open reading frame, or they see that like that it might have some expression, or it might be physically real, but they don't know what it is. So it's like I can basically take a protein, put it into AlphaFold, and reconstruct an entire three-dimensional image of what it might look like using what is considered an AI approach to solve structures. So basically what I'm showing here is that I could find what the radical is supposed to bind within this organism using an AI-driven approach. So yeah, it's pretty interesting. This organism does have soluble guanylate cyclase. I don't know if it's activated by nitric oxide, but it's pretty interesting that like you can, I thought, and this is why I'm showing it, is basically, I thought you can take like these hypothetical proteins that are shown in any database, and you can basically reconstruct them and show like how well they match to other proteins. Sometimes these AI approaches don't work as great as other softwares. Like I've shown before in the past, like antibodies don't always do well with AlphaFold, but sometimes they do. So it really depends on what you're looking at.
This looks like a TCR structure.
Yeah. So I was actually surprised by that. Yeah. So if basically on the top, what I believe to be the C terminus is, I believe, the soluble guanylate cyclase. But at the bottom is a 6-helix bundle, so I think that might be membrane bound.
Oh, okay.
Yeah, so it's interesting that like this organism has a membrane bound soluble guanylate cyclase. So that's something interesting, I think, to like these single-celled organisms. Yeah, usually with the mammals, you have something that like what I show on the right, and that's basically like a more soluble form. But yeah.
Okay, so AlphaFold 2 is free to download and use. Right?
Yeah. And possibly in the future, we can show the students like a tutorial.
Okay.
Yeah, basically you can access AlphaFold through Chimeric X, and that's downloadable from UCSF.
Did you find these hits by sequence homology or structural homology?
But yeah, so I started off doing pairwise alignments. So I basically,
Sequence homology.
yeah. And then basically when I overlay them, I can show that like how well they match, like I take known PDB files for soluble guanylyl cyclases that have been solved by like X-ray crystallography, and I can overlay that with the alpha fold structures, and they're basically almost identical.
Right.
Does that
For
make sense?
example, yeah, but AlphaFold uses known structures to infer what kind of structure any sequence
No,
will
yeah,
have.
it doesn't. So that's the idea, but so there's basically like two trains of thought. There's like ab initio structure determination where you're doing it from scratch, and then there's homology modeling where you're basically using known structures to solve a structure. So homology modeling is what you're referring to, and then ab initio is basically doing it from scratch. The AI driven approach is like a little bit of a black box to me though. Because it uses machine learning. And so, yeah, that goes into your question. Like I can easily just find a structure using pairwise alignment, and then I can overlay the different structures and show like how similar they are in sequence as well. So that's what I did by just finding this NOS within the features. Like I basically threw in like part of the sequence, found it, and then I can show by using Unicron how similar they are in sequence, and then yeah, basically confirm that it is like an ortholog within the species. So yeah, that's pretty much it. I just wanted to show like potentially what we how like NOS ties into other organisms, as well as like the significance of ROS accumulation and how it leads to feral apoptosis. And basically current evidence is showing that cancer cells evade feral apoptosis through metabolic reprogramming. I think it's an interesting topic that feral apoptosis, metabolic reprogramming, cellular immortality, and regeneration remains an unexplored topic, and no one has really tied all those together. I would think that there is some sort of relationship. And so, yeah, more or less, I just wanted to present on like what I've done before in the past, what I've studied, and possibly what I can help out with the students, and sort of my interest as well.
Since this talking affect
It's different based off of what T cell type of T cell you're talking about, or what sort of leukocyte you're talking about. It bigger question.
that's activation.
Yeah. So like macrophages naturally express like ions at higher levels, but there's always a basal amount of level of expression of NO within leukocytes. It basically it's different depending on which like T cell subset you're talking about. It can be pretty complex.
Like when you talk about metabolism reprogramming, like spiral process that they go oxidative phosphorylation versus glycolysis, which how do you switch? Glycolysis to oxidative phosphorylation, which way is
Oxidative phosphorylation.
usually when people talk about metabolic reprogramming, it's like a switch between glycolysis versus oxidative phosphorylation. ATP metabolism.
I see. So anaerobic versus
oxidative oxygen metabolism.
Yeah.
Glycolysis, you know.
Well, the body naturally, again, like produces enzymes, and also like it uses like glutathione and glutathione peroxidase to combat these the upregulation of ROS. But eventually, again, like if the amount of ROS exceeds what the cell can handle, then there is inhibition of certain iron containing proteins, and then it leads to cell death. But some cells, and there are some transcriptor factors that are upregulated when reactive oxygen species are produced. But it really depends on the cell. Like if it's an equilibrium, if the equilibrium is perturbed, then there is basically a phenotypic outcome. But it really depends on the cell type. Like some cells, again, like naturally produce catalase and superoxide dismutase at a higher levels than others. Yeah, it's not a paper I sent out, yeah, about NO and like T cells.
So the apoptosis would affect ion center and dye,
Yeah. So it can displace oxygen.
which is in mitochondria.
Oxygen? Yeah. So, yeah, mainly reactive oxygen species comes from mitochondria during respiratory respiration. So that's where like a lot of superoxide molecules are produced.
So the nitric oxide, this paper is talking about regulation of T cell in cancer by nitric oxide, but how that was working out. So it's something on the process, how this is regulate. How can cancer, it's like to me, cancer cell use this nitric oxide to try to protect itself, right? Is that what it says?
Yeah, like basal amounts are beneficial, but then increased amounts are bad. But again, like it goes to, like into this figure here. So you have natural mechanisms to combat this, and some cells are already differentiated enough that they have increased amounts of expression of like catalase, superoxide dismutase. Also upregulation of glutathione, cysteine anti-transporter. So essentially, like some cells are better equipped to quench the free radicals, and if they can, basically it leads to lipid peroxidation. Basically, the radicals will react with the lipids on the mitochondria and the cell membrane, and that leads to this sort of like cell death.
So the natural oxide try to manipulate, I mean, in the cancer cell, try to manipulate this process. So try to prevent this. Is that right? Is that so from I read, it seems like this NO, the cancer try to use this NO to protect itself somehow, to prevent something happening. Is that right? That this fatal process happen.
Yeah,
So I want to know the mechanism behind this, so that we can know how cancer cell works.
yeah,
Cancer
like
use
I
this NO to try to prevent. Yeah, I think maybe that's the idea. I want to get a conclusion.
yeah, the cancer cells take advantage of their environment for increased nutrient uptake. Like they'll uptake cysteine at greater amounts to produce glutathione and other methods to basically combat the reactive oxygen species within the cell, and that's why like they travel to lipid-rich environments like lymph nodes to increase the amount of lipids that they have and to basically combat the radicals within the cell or the cancer cell.
Okay, I think I need to read more about this because it seems like it's related to the T cell, cancer cell and T cell. Seems like it's there's some relation there.
Yeah, I can see how that can be very counterintuitive because if a cancer cell wants to avoid oxidative stress, why would it produce nitric oxide, right? That is quite counterintuitive, wouldn't you agree?
Yeah, why the cancer cell produces nitric oxide?
Because so the rate, the energy created from the average, so it's if you grow, like say T cells activation.
Yeah, so the reason why it's hard for
So
data to reconcile with the notion that nitric oxide contributes to oxidative stress and you want to avoid oxidative stress. It's because nitric oxide synthase has other roles as well. And one of them is by depleting arginine, but it's inhibiting the growth of T cells, for example. So that would be another role that is known for nitric oxide. So a lot of inhibitory immune cell types express nitric oxide synthase,
Right.
not primarily to produce nitric oxide, but to deplete arginine in the environment. So that's an alternative role for nitric oxide synthase. But yeah, expression of nitric oxide synthase is also not always a significant contributor to oxidative stress, right? Because even a cell that doesn't have this enzyme doesn't produce nitric oxide can produce tons of reactive oxygen species of other sorts just by breathing, so just by using that antibiotic.
We're interesting topic, yeah, just,
Yeah, I was gonna present on TCRs, but I think there's too many.
I tried based on you.
TCRs, so
Okay,
it was either TCRs or NOS.
okay,
Yeah, that's it.
thank you.
Okay, thank you. Free to go.
I need to.